Hello everyone, welcome to the third lecture of IMAT biology course. This lecture we are going to continue the cell unit, which is our second unit. We are going to almost finish it, and the next lecture is going to be more interesting. It's going to be a bridge between unit 2 and unit 3, where we deep down into the genetic material inside the cell. So from the cell structures, we are going to switch to genetics and inheritance, and we are going to do it inside the cell. But before coming to that part, today's lecture is going to be relatively easier, fun, and shorter. Today we're going to check. So as you can see, until now, these are the topics that we have covered. And today we are going to finish the cell membrane, its structure, functions, and transport across the membrane, which are easy topics, but quite important in the sense of the number of questions that they have in IMAT past papers. And next, we are going to start to learn about cell cycle and cell reproduction, but I plan to start this part in the next lecture. And before that, we are going to learn about viruses and bacteria and compare their structures and functions, because after pandemic, especially, comparison of virus and bacteria and specifically the virus structures have been started to ask on our exam. So now, if you're ready, we're going to start with cell membrane and its features. Until now, as you may remember, we covered about different types of cells: prokaryotic cells, eukaryotic cells, animal cells, and plant cells. And we learned about organelles. So cell membrane, you can count it as the last organelle that we are learning about, or you can completely take it as a next topic to make your revision on. So this image represents where we see the membrane structures within the cell. As we already covered mitochondria, and it's not shown here, but also chloroplasts, they have their own membrane. And at this point, I want you to remember about endosymbiotic theory. And if you don't remember what it is, just go back to the previous lecture. And besides these two complex organelles, we have membrane structure on lysosomes and obviously around nucleus, and around the cytoplasm as the cell membrane, as the outermost layer or membrane of the cell structure. Before we deep dive into the elements of plasma membrane, there is a model and a bit of history that you should know about when it comes to fluid mosaic model, the model that we describe the properties and how perfect the plasma membrane structure is for being semi-permeable and be selective and protective towards material exchange. So the fluid mosaic model of the membranes was first outlined by Singer and Nicholson. And thanks to this model, we are able to explain how passive and active movement can happen between the cells and their surroundings. We can explain cell-to-cell interactions and cell signaling. So these three articles you should know about, like you should have an idea that these are the things that we can explain with the fluid mosaic model. But we are not going to deep dive into these features of plasma membrane and how cells can use plasma membrane for different functions. These are the topics of histology and genetics. Once you start your med school, you are going to deep dive into these features. The fluid mosaic model describes the cell membranes as fluid because the phospholipids and proteins can move around with diffusion. The phospholipids mainly move sideways and they move within the layers, which we are going to explain with images as well. But moving with layer is like moving like this. So it seems like almost like they're flowing like a fluid, but they are not exactly fluid. And there are many different types of proteins that can be found on the bilayer membrane, and they can move around within the membrane. And when it's necessary, we can also find them in fixed positions. So as you can see, this fluidity can bring great variety in proteins and how proteins behave within the membrane. Another important point is the mosaic part of this model comes from the observation of the scattered pattern because of proteins that you're going to see on the image in next slide. Because of the scattered pattern, we called it as mosaic because it looks like a mosaic cake from the outside. And the main competence of the fluid mosaic model, you should know the last part especially. It's been asked. So when we talk about a fluid mosaic model or phospholipid bilayer membrane model, you should know that the features phospholipids, cholesterol, glycoproteins, and glycolipids, and transport proteins are included. Here we see a quite accurate image after years of research. Obviously, it didn't start like this. It was the images that we created, they were way more basic than this. But now, more or less, we are quite accurate. And this is how it looks like around the cells. This is the phospholipid bilayer structure. And usually on the images, unless it's stated otherwise, the upper part is the outer side of the cell and the lower part is the inner part of the cell. So you can take it like that. Here you can see the glycolipid structures, glycoproteins attached to the bilayer membrane. There are some peripheral proteins, some integral and channel proteins. If you don't know what do they do, you don't have to worry. We're going to briefly cover them. But the good thing is IMAT doesn't go into detail about membrane proteins and their specific names or specific functions, but it's better to have an idea about them. Especially channel proteins are our besties. We are going to learn lots of things about them and how useful and lifesaver they are. But let's hold on the proteins for a while, because first we are going to cover the phospholipid bilayer proteins and what does bilayer mean? Like, how do we create this double layer, bilayer structure? So phospholipids form the basic structure of the membrane, and they are formed by a hydrophilic phosphate head bonding with two hydrophobic hydrocarbon or fatty acid tails. So for our exam and in general, in order to understand plasma membranes, it's important to understand this structure. You should understand hydrophilic means water attaching, water loving part of the structure, and it contains a phosphate head, and the tails are hydrophobic or fatty acid tails. They don't like to bind with water. They avoid water if that's possible. And this double property of one side loving and attaching to water and other side doesn't like to attach to water, this double property gives perfect features to bilayer membrane. It allows us to export or import different materials. And remember, every kind of chemical that we are talking about inside our body, they are either polar or non-polar, or with other words, they are either water dissolved or non-water dissolved. And at this point, if you have no idea what I'm talking about, I'm sure that the chemistry course lectures are going to be helpful for you, because in chemistry lectures, obviously, we are going to break down what a polar structure is, what a non-polar structure is, how do they interact with water, how we break the bonds or form the bonds. If you don't feel confident about these topics, you should check my chemistry course in future. Moving on, by the way, we call this property as amphipathic. So having that we have both hydrophobic and hydrophilic parts in a structure is called amphipathic. And this amphipathic structure, that having these two features, when they are placed in water, the hydrophilic heads, they are going to turn towards the water, just like in this image. And hydrophobic tails, they are going to stay away from the water, or vice versa. If we put them inside an oil the tails will stick to the oil part, and the heads are going to avoid the oil part. Here is it's not entirely a plasma membrane structure, but hypothetically or experimentally, we are talking what would happen if they were shaken with water, and how would they change their direction. And at this point, I like to give an example. Probably this is something, especially the girls will understand better. I'm sure that if you're using makeup, until now you heard about micellar water or micellar makeup remover. And micellar means that the material inside your makeup remover, it has both hydrophobic and hydrophobic parts, so that it can remove both water-based makeup and oil-based makeup. So that's why we call them as micellar water or micellar makeup remover, because they have this double structure that can work on different kinds of makeup. Here also, this is another possible shape that they can take when there's sufficient concentration of phospholipids in the two layered structures, that they can form sheets like this. And these sheets are going to be called phospholipid bilayers because of this dual structure with heads and tails, heads and tails. And that's why we tend to call the membrane as a phospholipid bilayer membrane, because this sheet-like structure is how we find them around the cell cytoplasm. Furthermore, the two layers of phospholipids are loosely held together by weak hydrophobic interactions, and the amphipathic properties result in phospholipid bilayer acting as a barrier to most water-soluble substances. Imagine cytoplasm is mostly consists of water, and depending on the environment, the cells, they have to either excrete the water inside them or try to not to lose the water inside them. So this bilayer structure and hydrophobic interactions within the membrane is going to be a barrier to not to lose the water. Or think about the other hand, we are like, as the cell, we are trying to protect our water, so like water cannot escape from the barrier that we create with the hydrophobic interactions. But in the meantime, you have some sugars, amino acids, proteins that you have to keep within the cell. So you want to also keep them to yourself, because those are your nutrients, or you're going to build something with it, some another reaction with it. So you have to keep them. So this also ensures that water soluble molecules can stay inside the cell without leaking out. So now, until now, we covered the amphipathic property of phospholipid bilayer membrane. Now we are going to talk about the other features of the membrane. As the beginning, we said there are four main features to know about the model. So now we are going to move on with cholesterol. Cholesterol is not asked, until now at least, they haven't asked about cholesterol in detail, but here and there you're going to see about cholesterol, so you should know what it is. And also, as a future med student, I think it's important to have an introduction with the cholesterol, because the way we use cholesterol in daily language and the way we use it in medicine is quite different. Usually in daily language, we tend to announce cholesterol as the evil, as the bad thing, or the starter of bad diseases and bad things inside the body, but that's not entirely true. So you have to know how we use cholesterol around the cell membrane. So it's a lipid structure, belongs to the steroid group. It is amphipathic, and the majority of the cholesterol molecule is hydrophobic, and therefore it can attach to the hydrophobic carbon tails on the membrane. So if a material is hydrophobic or nonpolar, it's going to attach to the tail part of the bilayer membrane. Or if the molecule is polar and likes to dissolve in water, they are going to attach to the heads of the bilayer membrane. I think that is pretty easy to imagine, but I wanted to remind you that when you're in doubt, just check about their polarity. Are they water soluble or not? If you have to figure it out. And this is how one molecule of cholesterol look like. And without going into too much detail, because there are lots of things that we can talk about cholesterol, how we use cholesterol around the plasma membrane, how we use cholesterol in the bloodstream around the vessels, how we use intake or outtake cholesterol through nutrients digestion, how we like what is the role of liver when it comes to handling cholesterol, what are the proteins that carry cholesterol. Like there are lots of things to learn about, and it's quite interesting. But as beginning, I want you to know that like cholesterol is not the enemy, and it naturally exists within the structures of our bodies. So let's check the mammalian membranes and what does cholesterol do with them. So remember, plasma membrane is fluid or fluid-like, and the components are relatively free to move around. And this fluidity of the membrane has to be controlled. Of course, like we want some fluidity, we want movement to some extent, but obviously we cannot have too much movement that we leak out the materials or lose them around. So this needs to be controlled. So too much fluid is not good, and not fluid enough is also not good. And cholesterol has a role in that. It helps with the regulation of the membrane fluidity and permeability. And the interaction between cholesterol and phospholipid tails stabilizes the plasma membrane at higher temperatures, especially. Cholesterol molecules bind to hydrophobic tails of the phospholipids, and the stabilization occur, and therefore the phospholipids, they stay together, they get packed together because of this interaction. On the other hand, at colder temperatures, cholesterol, still the same guy, it can increases the fluidity of the membrane. Do you see this dual role? Like in the hotter temperatures, it avoids it being too fluid, or it helps with the packing of the plasma membrane. But at the same time, is if the temperature is cold enough, it increases the fluidity of the membrane and avoids crystallization or cell becoming too rigid. So it's great. It's really helpful. In fact, certain amount of cholesterol is quite important for our skin elasticity, to avoid wrinkles, the signs of aging. So especially, I'm telling you out there, if you're avoiding cholesterol or animal-based fat resources in your diet, you should rethink again. And this is not the only reason, but I'm just trying to include some extra informations because you're a med student candidate, and probably it can be more interesting than just reading out the slide. And furthermore, the mechanical strength and stability is possible thanks to the cholesterol. But this is it for now about cholesterol, because we have to move on and stick with the curriculum. Now we are going to talk about the membrane proteins, because membrane proteins are relatively more important for IMAT exam. Again, as the plasma membrane, the phospholipid bilayer, it has some essential functions. For example, to control the substance exchange. And there are some other functions that proteins can do. Plasma membranes are called globular proteins, and these globular proteins, they are a huge family of proteins, okay? And we can have two subcategories of globular proteins. First one is integral. They are partially hydrophobic, and therefore they can attach to the phospholipid bilayer. And peripheral proteins, these guys are quite hydrophilic, and they can temporarily attach to the surface of the integral proteins. So integrals are more permanent proteins on the membrane, and peripheral ones are like we can attach them or unattach them depending on the need or the current function happening. Furthermore, let's talk about what these membrane proteins can do. Because, okay, the membrane on its own is responsible with being semi-permeable and provide protection and control the substance exchange. But what do these proteins can do? One of the important functions is transport. The transport proteins can transport ions and polar molecules to travel around the membrane. The transport proteins can be further categorized into channel proteins, which have a pore inside their structure. Carrier proteins. Carrier proteins are more like a hair tie or some sort of that you can think of, for example, a chain that change the shape, open and close. Because channel proteins, they don't change their shape because of the pore that they have inside, but the carrier proteins, they are the ones who gets opens or close, which we are going to see on an image. And this change shape, it has something to do with the energy consumption and the specific substances that they can carry, which we are going to learn further. And obviously, each transport protein is present specific for a particular ion or a molecule. Not every protein can transport every kind of molecule. Furthermore, we have receptor-like receptor group of proteins. They are meant to bind to the peptide hormones, for example, insulin, or neurotransmitters or antibodies. They are there to receive other productions from the other parts of the body and act accordingly. Immobilized enzymes are a subgroup of integral proteins, and the active site of them should be exposed on the surface so that the active site can bind to whatever the substrate that is meant to bind. And cell adhesion, it allows tight junctions. Junctions are packed formation with very complex features in between the cells. It has to do with stability. It has to do with being packed in specific locations, and it has to do with material exchange and communication. When we say tight junctions, there are lots of further things to talk about, but this is not an IMAT topic. This is actually a med school topic. Cell-to-cell recognition can happen thanks to the glycoproteins because they act like they have a sort of identity, different shapes, so they can be cell markers or antigens, or they can be simply useful for cell-to-cell recognition. For example, cell-to-cell recognition and glycoproteins are essential when it comes to determining the ABO blood group antigens and act accordingly, because obviously if you are looking for a blood transfer, you should be able to identify the glycoproteins on the red blood cells of the patients. And luckily, we are going to learn about ABO blood group determination and what do antigens means, what do antibody means. We are going to learn all of that because we are responsible about understanding the ABO blood group and the heredity of the blood groups. Here, this image, I think it summarizes quite well all the proteins and subtypes of the proteins that we have talked about so far. Now, let's talk about types of transport. And obviously, we are going to learn how we do them, what kind of materials we can transport, how do we transport, what kind of proteins we need through the transport process. So for IMAT exam, we are responsible with passive transport, active transport, and osmosis. And we are going to cover all of them in this lecture. Starting with passive transport. Passive transport is the movement of ions or other molecular substances within the cells along the concentration gradient. Now, this is quite important. When it's passive transport, it has to happen through the concentration gradient. So, what does this mean? Maybe you don't know the concentration gradient. It means that the molecules have to travel from the higher amount of the molecules to the lower amount of the molecules. So this is the direction from the higher amount of to the lower amount of. And because of this concentration difference and because of this movement from higher to lower, we do not have to use energy. And that's why we call it as passive transport. What I just said was super important. What are the types of passive transport? How do we make it happen? Actually, we can say there are three subtypes of passive transport because essentially osmosis is also a passive type of transport. And when we say passive, again, we mean there is no extra energy consumption for this transport. When we say active, we mean that we have to use ATP, we have to use energy to make that transport happen. So we can say that simple diffusion, facilitated diffusion, and osmosis, they are all types of passive transport. And as you can see on this image here, simple diffusion happens without any helper protein through the cell membrane. When we say facilitated diffusion, we mean that there is a channel or carrier protein that helps with this transport. It actually allows to the pass of the material. And osmosis is specific for water molecule. It's the movement of water through a semi-permeable membrane, which we are going to cover later. Now, there are some further informations about these subtypes of passive transport. Simple diffusion happens through the plasma membrane, through in between the phospholipids in the plasma membrane. The exact definition is, like the book definition is, the net movement as a result of the random motion of its molecule or ions of a substance from a region of its higher concentration to a region of its lower concentration. So what does it mean? We move molecule and the net movement of the molecule as the result of a random motions through the regions, and it has to be from higher concentration to lower concentration without using any protein, without using any ATP. So when there is this concentration gradient, when there is, okay, here in the middle of my hands, we have the plasma membrane, and here we have the higher concentration area, and here we have the lower concentration area. So from higher to lower, when the movement happens, it actually happens thanks to the kinetic energy of the molecules and ions, because as we are going to learn in chemistry course, every molecule, they have their own amount of movement and spinning in the environment that they are present. So when there is this concentration difference, thanks to their own movement, thanks to their own molecular movement in the environment, this transport can happen. And here you see an image representation of it. From left to right, time passes. And as you can see, from higher concentration to lower concentration, in this case, the extracellular fluid, like the extra cell part, has the higher concentration, and the cytoplasm part has the lower concentration. And through the time, you can see the number of molecules changing in both sides. So the rate, which is a bit extra information, but the rate of this transport depends on the amount of the difference in the concentrations, the temperature, the surface area that we have. And it also depends on the type of the molecule, because large molecules are going to diffuse slower, uncharged molecules are going to diffuse faster, nonpolar molecules are going to diffuse faster, and polar molecules, it's kind of harder for them to pass through the hydrophobic part of the membrane, but if they are small enough they can diffuse in very low rates. Now we continue to the second subtype of the passive transport, facilitated diffusion. As you may remember, in facilitated diffusion, we have to use a sort of protein. It can be channel protein or carrier protein. So, what do we transfer with facilitated diffusion? First of all, large polar molecules like glucose and amino acids. Here in this all transport topic, the molecular names, like the molecular names, the examples, specific examples are important for our exam, because I remember there were questions as which one of the molecules can be transported through facilitated diffusion. And if you think that glucose is too big and polar to be passed, to be transferred with facilitated diffusion, you're wrong, because we can actually transport glucose with facilitated diffusion without using any ATP. But there are some other conditions that we might have to use energy in order to transport the glucose. For example, if there's no concentration gradient from high to low, if we are trying to do the opposite from low to high, then in that case, we might have to use ATP. However, again, when I give molecule examples in this particular topic, please pay attention. Ions such as sodium and chloride, they can also be transferred by facilitated diffusion. And at this point, we are going to get to know channel proteins and carrier proteins a bit. Channel proteins are the ones with the pores. The pores are water filled. They allow to the pass of the charged substances like ions. The diffusion of these ions, it doesn't happen freely, but usually most of the proteins, they are gated. They are sort of closed. So the part of the channel protein on the inner surface of the membrane can move in order to close that pore, and when it's necessary it's going to get opened, so it gives the control to the cell whether to transfer that ion or not. By keeping close the gates of those proteins, we can actually control if we want to transfer them or not to transfer them. Here you see an example of channel protein structure. And as we just said, we can transfer sodium or chloride in. Here you see the movement of the sodium. So when it's open, the sodium can move freely, but when it's closed, it's not going to. Now let's talk about carrier proteins, because they are a bit different than the channel ones. They don't have a fixed shape. Instead, they switch in between two shapes, as you can see on the image. So initially, what happens, the binding side of the carrier protein has to be open at the one side of the membrane. It's not going to be always from exterior to interior. It can be also from interior to exterior. Remember that. It depends on the concentration difference. It's not always going to be towards inside the cell, because sometimes we have to export materials from inside the cell to the outside environment. And again, this since this is a type of passive transport, the direction of the movement will depend on the concentration gradient, and the net diffusion of the molecules or ions will occur down a concentration gradient, and the shape will change in order to make this transfer. And as you can see at the first, the upper part of the protein was open, but in order to transfer the molecule, it changed its shape and got opened downwards to release the molecule to the cytoplasm. Now, another type of passive transport, osmosis. As we already know by now, all cells in our body are surrounded by a cell membrane, and cell membranes are partially permeable. The water is the cool guy. It has its own VIP card through this partially permeable or semi-permeable cell membranes. Water can move in or move out by osmosis, and osmosis is the diffusion of water molecules from dilute solution to more concentrated solution. So what did I say? Dilute means it has less concentration, and the movement happens from dilute solution to the more concentrated solution. Now, what I told can be tricky at this point. I like to clarify. Until now, we have talked about the abundance of the other molecules in the environments. Now, when we talk about water transfer, when we talk about osmosis, when I say dilute solution, you should imagine a solution with more water molecules. So it's water abundant, but it's diluted, because in chemistry, when we say dilute, it actually means that like we have less material inside the solution. But since it's diluted, it actually has more water and less other materials. So it's important to not to confuse at this point. So we are passing from more water abundant area to less water abundant area. The concentration gradient logic is the same. Now we have an experiment example here. We see a solution with the semi-permeable membrane, and we observe the movement of the water. So now on the left side, it's sugar solution. It's called like concentrated or sugar solution. And on the right side, we have less concentrated or diluted solution. So it has more sugar and less water on the left side, and on the right side it has more water and less sugar. Now I don't want you to confuse. We are talking about the movement of the water, so you should pay attention to the abundance of the water. The movement will happen from the more water concentrated area to the less water concentrated area. Okay, I have to make this word trick here to make you understand. The water movement happens from more water abundant area to less water abundant area. So when I say diluted, it's actually more water abundant, and when I say concentrated, it's actually less water abundant. Is important. And by the time as they the thing about osmosis is not going to continue all the time. Once the both sides of the permeable membrane are equal in the sense of water concentration, the transfer will stop, obviously. Now we have some definitions about solutions, which I like to explain in this context. So when we say in biology and in medicine, when we say hypertonic solution, it actually means that the hypertonic solution, it has lots of solute or foreign materials inside the mixture or inside the solution. So it's less water abundant comparing to the internal environment of the cells, because like the cells, they have water inside them, their cytoplasm, no matter what. But if we put them in this hypertonic solution, which is rich in some sort of molecules but not rich in water, the net movement of the water will be from inside the cell towards outside the cell. So therefore the red blood cells, they are going to lose water and they're going to get shriveled. This is the English word that you should know. And the net movement of the water will be towards outside. If the solution is isotonic, it means that the water concentration, the water potential is equal in this environment and inside the red blood cells. So there isn't going to be a net movement of water, and it doesn't mean that there is a zero movement, like water movement can happen, but it's going to be equal. So therefore the net will be zero. Water can pass through the cells, but also is going to pass in to inside and also from inside to outside. So the net movement will be zero, and the red blood cells, they're not going to lose any water, so there will be no change in their shape. And the third possibility is being in hypotonic solution, where water is more abundant in the outside environment and it's less in the cell cytoplasm. So therefore, red blood cells, they have lower water potential, and the net movement of the water will be from outside to the inside of the cells. And therefore, since these cells are intaking water, they are going to swell. And if it happens too much, like if they stay in hypotonic solution in too long, they are going to burst because of the amount of water that they are trying to absorb. So these solution environments are going to be important when it comes to cardiophysiology, when you want to understand hypotension, hypertension, what's going on in the bloodstream of the patients, and hopefully you will remember. Now we have further examples to the facilitated diffusion. I specifically wanted to include these examples because until now, in the physiology part about these axons, we had one question. And even if it's just one question, still I want to make all topics relate to medicine somehow, because that's majority of the exam papers have done so far. So the axon is a part of the nerve cell or neuron. It has a long, narrow tube structure, and it transmits nerve impulses, and it can transmit these nerve impulses thanks to the sodium and potassium ions being moved across the axon membrane. So through the axon membrane, we have facilitated diffusion for sodium and potassium. And this is a representation of it. You can pause the video to check the image further. And now we are going to move on with the active transport, what it is, what are the conditions, what do we transport with active transport. Active transport, by definition, is the process that involves movement of molecules from a region of lower concentration to a region of higher concentration. So we are working against concentration gradient, and now it's like in previous passive transport types, it was our supporter. We were benefiting from that, but in this case, we have to work against it. So it's our challenge. And that's why we need external energy. We have to use external energy. In this case, at the cellular level, we are going to use ATP. So for active transport, ATP is not the only thing that we have to use. We have to use carrier proteins, and each carrier protein will be specific for particular molecules or ions. And it's true facilitated diffusion. It's very similar to facilitated diffusion using carrier proteins. However, in this case, we are working against the concentration gradient, and therefore we require the energy. And this energy is going to be used to change the shape of the carrier protein so that we can transfer the molecule. The specific way we use ATP, the specific process is called hydrolyzation or hydrolysis of ATP to release its energy. Now you can see how it looks like. We're working against concentration gradient. So if it's higher inside the cell and lower outside the cell, and if we want to make the transfer happen from the lower part to the higher part, then we have to use energy to change the shape of the carrier protein. So some body examples, human body examples for active transport are phagocytosis of bacteria by macrophages, movement of calcium ions out of cardiac muscle cells, transportation of amino acids across intestinal lining inside the small intestine, secretion of proteins like enzymes, peptide hormones, antibodies, and functioning of white blood cells to protect our body because there is a release process of white blood cells, and in that release, which is a very complicated process that you don't have to know about, but to make it happen through the foreign body abundant areas, we have to use active transport. Now, as you can see, axons are quite hardworking and interesting parts among the inside the neurons. And previously, we mentioned that they use facilitated diffusion for sodium and potassium, but also they can use active transport if they are working against the concentration gradient. So the integral proteins among the axons, they can be used as sodium potassium pumps, and for every how can I say, every pump, like every fire that we have to do this, we have to move 3 sodium ions out of the axon and 2 potassium ions into the axon, and we use 1 ATP molecule per cycle. Here you see a very simplified representation of it. It's actually a quite complicated process, but you have to know to some extent about how the neurons work. A similar process is happening with the calcium ions through the muscle cells, and we are going to learn about the works of the muscle cells, how they create movement, and also for neurons we have to know how do they create the messages through the nervous system. So we are going to cover briefly in the physiology part. Finally, we are going to learn about endocytosis and exocytosis. They are also called as bump transfers or pack transfers because we are transferring high amount of materials at one time. So therefore, like they are being used if we need the transfer of big amount of materials. And it's the endocytosis is the process of plasma membrane engulfing the material. Maybe you remember from the previous lecture, we said that the plasma membrane can create an extra sac, extra endocytic vacuole around the material that it has to transfer. And there are two types of it. It can be phagocytosis or pinocytosis. Phagocytosis is the bulk intake of the solid materials, and pinocytosis is the bulk intake of liquids. And there is an opposite transfer of it. Endocytosis was to intake, and exocytosis from the X, you can imagine exit. So exocytosis is to make the material exit the cell, and we have to transport them out. We have to transport out enzymes, hormones, or other waste materials. And it happens thanks to the efforts of Golgi, checking the material and make sure that they are vesicled They are in the sac and they move towards the cell membrane surface, and then they release their content to the outside of the cell. So for now, regarding the cell structures, that was it. We covered the cell, types of the cell, the organelles of the cell, cell membrane, and transport through cell membrane. We are going to finish this lecture after we learn about virus and bacteria and compare them, because as you may remember, I told you that IMAT biology loves comparing questions. So after we learn briefly about them, we are going to compare their properties. Before we dive into it, here's a small recap. I want you to remember that prokaryotes are single-celled organisms belonging to the domains of bacteria and archaea. Prokaryotes are way more basic and smaller cells comparing to the eukaryotic ones. They do not have nucleus and they do not have any membrane-bound organelles. They have only ribosomes, if you remember, 70S ribosomes. All prokaryotic cells are encased by cell wall, and many of them can have accessories like capsules or slime layers. And also, furthermore, some of them can have flagella or pili for motion, or they can have fimbria to create cell adhesion, or a sex pili for DNA exchange. Now you see a prokaryote type, which is bacteria cell on the image. Bacteria are single-celled prokaryotic microorganisms, and they are abundantly present in our world. And they like to find hosts, if that's possible, but also they can live in nature like soil and water. They can be both beneficial or harmful for health of humans, plants, or other animals. And on the other hand, the virus, we define virus as a cellular structure that requires a living host to survive. Normally, it doesn't have a cell, it doesn't have cell features, but it has some other structures that makes the virus attached to a living host and start to act like a living thing. And its main goal is basically to cause trouble. It likes to create illnesses for the host. It likes to attack to the immune system. Bacteria are alive for sure, but we still have ongoing discussions about viruses, whether they are living or non-living things. Here's a introductory image. It's actually from a video that I like to include in my external resources about bacteria. So we have the cellular build, cytoplasm. It shows life processes. It contains genetic material. It has cell membrane and cell wall, and it can feed from the environment. So comparing to viruses, bacteria can be independent and can be present independently in the environment as well. So let's see what kind of troubles can bacteria can cause human body. Here are some examples of bacterial infections. If you want to stop and read the names to get familiar with them, please do so. But before we finish with the bacteria, I would like to mention that not all bacteria are harmful to humans or plants or animals. Some of them are quite beneficial in different ways, and I wanted to give you the examples so that you're not going to get surprised if you ever see them. For example, lactic acid bacteria or lactobacillus can help us convert milk into curd, and we ferment food products thanks to streptococcus and bacillus. They help in digestion and improving the body's immunity system. Actinobacteria, proteobacteria, bacteroidetes. These are the guys that can be present abundantly. They are being used in the production of antibiotics, which is quite hilarious. And we use these bacteria in the treatment of and the prevention of other bacterial infections. In fact, the microbiome, the bacteria, the millions of bacteria inside our guts are quite important to be able to digest with no problem, to have healthy mood regulation, and to not to have other disease constantly. So you should also take care of the good bacteria inside your body. Now let's talk further about the viruses. Where a virus, we can say that they are infectious particle, and by infectious we mean that they sneak into the other living things. They always try to find a host cell or a host organism. They can be made up of DNA or RNA genome, and this genome is protected inside a protein shell called capsid. Some viruses, they have another external membrane called envelope. Viruses are very diverse when it comes to shapes, structures, genomes, and how do they infect their hosts. They can reproduce by infecting their host cells and reprogramming their cells to make them hack. It's like hacking a laptop. That's why we call them virus for laptops as well, because it's a similar mechanism. They reprogram the cell, they change the genome and the process inside the cell so that the cells start to work for the virus under their command. The viral structures that you should know about, for example, this is a bacteriophage and this is human immunodeficiency virus. As you can see, they can be very different in shape, but the structures can be similar in the sense of function. They have capsid where they protect their genome. They can have DNA or RNA. They have envelope. This one has envelope. This one on the left, it doesn't. And they can have envelope protein and enzymes as well. Again, this is a bit more summarizing, recapping, but let's talk about one more time about the properties of the viruses. They are non-cellular organisms and they are enclosed in a protective envelope and they have their own genome. The presence of the spikes help in attaching the viruses to the host cell. It's a very complicated process, by the way, but we are not going to learn about it. They do not grow. They do not show any metabolites. They do not show any life functions that we learned at the beginning of the cell unit. They do not show these signs on their own. But once they find a host cell, they reproduce. Again, they can have either DNA or RNA nucleic acid. And that's why, because of this special case, they're both considered as living or non-living things. Again, when we say DNA virus, RNA virus, I don't want you to get confused. This is about the type of genetic material that they have. If you don't know anything about DNA or RNA, don't worry, because in next lecture, we are going to learn about DNA and RNA. These are the further categorizations of DNA viruses. I wanted you to see their shape and structure. And these are the further subtypes of RNA viruses. If I were you, I would pay attention to the names of the example viruses as well. For the reproduction, there are actually multiple ways to reproduce for a virus, but we are going to learn about lytic cycle because it's the most common one. Majority of the viruses reproduce with lytic infection. So a virus penetrates the host cell during this infection, it replicates and causes the cell to lyse or explode. So the attachment of the virus happens to the host cell. The entry, the injection of the genetic material to the host cell happens. And then replication starts. The replication is in favor of the virus because virus with its own genome, it takes control of the host cell metabolism. Now the cell is the factory of the virus to reproduce. And assembly happens. It's a complicated process because basically the genome of the virus take over the genome of the cell, and the production of the proteins inside the cell happens under the command of the genome of the virus. And furthermore, the release happens. The virus enzymes induce the host cell to burst so that the further viruses that has been produced inside they can be spread to the environment. Here are some examples of viral infections. The common cold, for example, quite important to know that it's caused by viruses. Hepatitis A, B, C, D, E, like it's easy to know about hepatitis, so you can check. Pancreatitis, there are different viruses that cause gastroenteritis, and you can stop and check the further examples here. And obviously, coronavirus is RNA virus. And here's a recap image for virus structure. Again, this is from the same video that I just mentioned for the bacteria part. They have simple morphology, they have capsid, they contain genetic material, they do not contain cytoplasm, they do not show any life processes. Now, finally, we are going to take a look at the table that I created to compare their properties, and we are going to finish this lecture. So you can check their definitions. Since we just covered them, I'm not going to read that part again. Bacteria can have ribosomes. Virus, they do not. Cell wall is present in bacteria as they are prokaryotes, obviously. Virus, they do not have cell wall, but they have capsid. Bacteria are considered as living organisms, and virus, they cause infections and they also cause discussions. None of them have nucleus. The number of cells can vary. Usually bacteria are unicellular, and viruses, they do not have cell to begin with. And the structures, the bacteria can have DNA and RNA. Again, the genetic material is naked, so it's not protected with the protein shield, but they have DNA or RNA. And the DNA or RNA of the virus is enclosed inside a coat of protein that we just learned about. The reproduction is binary fission. We learned in lecture 2. It's a form of asexual reproduction. On the other hand, virus, they have to invade a host cell and take over it in order to copy themselves. We don't say they reproduce, but we say copying their cells. Treatment is possible for bacteria with antibiotics. On the other hand, antibiotics, they do not work on viral infections, which is super important to know, both for medical and non-medical people. So you cannot treat common cold with antibiotics. And vaccines are the most efficient way to treat the viral infections. And sometimes we can develop antiviral medications, but it's super hard to develop them, and the response to them are not as efficient as to the response to the vaccines. So when infection occurs with bacteria, it's always starts and generally happens in localized way. But when virus infects an organism, it's a systemic. It's all over inside the organism. We can have some beneficial bacteria. However, viruses, until now, we didn't see any good from the viruses, so we do not consider them as beneficial. And regarding the size, bacteria are larger than viruses. Now we are at the end of the lecture 3. Here are the things that we covered so far. At the last lecture of Unit 2, which will be the next lecture, we are going to learn about cell cycle and mitosis. And then we are going to take a look into DNA and RNA, the genetic material inside the cell, which will be our connecting point to the Unit 3, which is about inheritance and genetic materials. I hope you enjoyed this lecture. Please check the extra resources and links that have been provided with and make your revisions. And we will see each other in the next lecture. Bye.